Hello everyone, my name is Carl Larson from RMCG, part of the Soil Wealth and Integrated Crop Protection team. And today's webinar, we're going to be delving into lettuce viruses and a real focus on managing those and identifying them properly to reduce crop loss and damage to the hip pocket. We're really pleased today to be joined by Cliff Kinoti from Agriculture Victoria, who was part of the broader area-wide management project that's just been recently completed and led by the Queensland Department of Agriculture and Fisheries. Before I do hand over to Cliff, the main purpose of today, we want you to come away with some really practical tips and tools. So you'll notice on Cliff's slides, there's lots of photos and lots of practical information, which is really grounded in cutting edge and best practice research as well. If this is the first webinar that you've attended, you do have the control panel on your screen there where you can type questions. They'll come straight through to me and we can put them to Cliff at the end where we've got time for a Q&A. There's also a handouts panel that has the two guides, one for bacterial disease, one for viral diseases, which are hot off the press. So feel free to click those and download them. There's also a host of other fact sheets that are relevant to the lettuce viruses that we'll be covering today. All right, so Cliff, welcome. You're going to be talking to us about managing lettuce viruses. We're really looking forward to what you've got to say. We'll hand over to you. Thanks.
Thanks, Carl. Thanks for this opportunity. So today I'm going to talk about managing lettuce viruses for profitable vegetable production. And this is some of the outputs from the area-wide management project that concluded recently. And when we talk about lettuce viruses, sorry, not one. So when you talk about lettuce viruses, there are more than 10 viruses that infect lettuces. This is what we found out through the project. And out of these 10 viruses, we identified the top 5 as the main viruses that infect lettuces. And these are the viruses that I'm going to focus in this talk. So we're going to start with lettuce mosaic virus, which is associated with lettuce mosaic disease. And here's an electron microscope image of how the virus particle looks like. Symptoms expression of lettuce mosaic virus in lettuce depends on the strain and the timing of infection. And lettuce mosaic virus can infect several vegetables apart from lettuce, such as endive, peas, spinach, chickpea, and mustard. And it's also found in weed hosts such as chickweed, salthistle, prickly lettuce, wild lettuce, and other weeds. And these weeds are an important source of the virus to infect lettuce crops. So in terms of spread of lettuce mosaic viruses, seed is the main mode of transmission of the virus, but virus is also transmitted by many aphid species such as the green peach aphid, melon aphid, your potato aphid, and south thistle aphid. Aphid transmission of lettuce mosaic virus by aphids is a non-persistent manner where the aphid pick the virus, and after short feeding periods or probing on an infected plant, and it can be infective for a period of 1 hour to 2 hours, and this is where the aphids, while it's flying across the crops, can infect your plants. Mechanical transmission of lettuce mosaic virus has been demonstrated, but it's a minor mode of transmission, especially in fields. In terms of disease symptoms, lettuce mosaic virus is characterized by a mosaic or mottling pattern on younger leaves. And also we find vein clearing and browning followed by partial necrosis or death of inner leaves. Other symptoms include leaf margins that are ragged or jagged in appearance, as you can see in the bottom picture here. And also some of the young leaves may become twisted and partially folded. Early infection of lettuce mosaic virus results in severe stunting, and plants fail to form hearts, as you can see in this picture here and also this one here. So the next lettuce virus that's also important for lettuce production in Australia is lettuce necrotic yellows virus. And according as its name suggested, it's associated with lettuce necrotic yellow disease. The disease is observed mostly in warmer months and can cause huge losses. Lettuce mosaic virus also can infect other vegetable hosts apart from lettuce. That's your endive, chickpea, garlic, and lupin. And also it's found in a lot of weed hosts, which act as an alternative host for this virus. In terms of the source and spread of the virus, the blackcurrant or the sow thistle aphid is the most important vector of this virus, and sow thistle are the most important breeding and feeding host. So lettuce is not a preferred host for this aphid, and this aphid is persistently infected with lettuce necrotic yellows virus, which means the aphid is infectious for life once it picks up the virus. And transmission of the virus by the sow thistle aphid occurs during migration from one crop to another and with feeding periods of 1 to 5 minutes. And this explains why it's a huge problem and it can cause massive outbreaks. The other important aphid for lettuce necrotic yellows virus is the current lettuce aphid, and this is an important pest of lettuce and chicory and the whole family of lettuce type of crops. It mainly colonizes the lettuce hearts and rosettes, which makes it very difficult if you have to use chemical controls to manage the aphids. And also this current aphid can also transmit other viruses that are aphid transmitted, such as cucumber mosaic virus or alfalfa mosaic virus. In terms of disease symptoms, they differ depending on the age of the plant, and initial infection of the virus is characterized by dull and bronze leaf symptoms. And then the plants become yellow, necrotic, stunted, and often have twisted leaves. This is followed by wilting, flattening of plants, and finally the plant might die. And early infection prior to heart formation might result also in internal necrosis and death of the plant. The next important lettuce virus is lettuce big vein virus that causes lettuce big vein disease, and this is caused by two viruses. And the first one is the Mirafiori lettuce big vein virus and the lettuce big vein associated viruses. So one virus can cause this disease, or both when they occur in a plant can also cause this disease. And as you can see on the right, is the two electron microscopy image of the viruses. So the lettuce big vein disease can occur all year round, and it's more prevalent during low temperature where it's wet and in heavy soils. Its host range is mainly the lettuce family, that's the lettuce, endives, and radicchio. But it's also found in other weeds similar to other viruses. That's in capeweed, you can find it in prickly lettuce, in nightshades, and also we found it also in sow thistles a lot. In terms of its transmission, transmission of the virus is associated with cool, moist temperature, and also it's transmitted by the fungus Opidium virulentus. So this is one of the most unique viruses that are transmitted by fungus. And the fungus in itself does not cause any damage to the lettuce, but the spores that the fungus produces move through wet soil and transmit the viruses to lettuce roots. The two viruses that are spread from infected lettuce and to nearby crops, and this is how the virus spreads across the fields. And one thing about this fungus, it's very stable in soil, and it can remain in the soil for up to 25 years, causing infection when there's continuous cropping of lettuce. Infected plants and also contaminated equipment is one way where contamination by this fungus can occur in a field and further cause spread of the disease. And here are some of the images of this fungus in roots of lettuce and some of the spores formation that are released to spread the virus. In terms of symptoms, they vary depending on the age and also the variety of letters, but generally the disease symptoms are characterized by thickening of the veins. And also we find a lot of blistering and malformed heads and standing, which is common with this disease. There's also severe plant stunting if infection occurred during the early stage, or reduced formation of lettuce heads. Later infection may only just show the big vein symptoms in leaves, as you can see here, where the head is formed, but you can see a bit of symptoms of the lettuce big vein leaves. One thing about infection with lettuce big vein disease, some of the plants, lettuce plants look normal and healthy, but the storage shelf life is short compared to other healthy ones. So the next most important virus is tomato spotted wilt virus, and this is an important virus across many vegetables, not only for lettuce. It was first reported in tomatoes in Australia in 1950 in the Werribee region, and since then it's found across the world. And here is an electron microscope image of how the virus particle look as cylindrical particles. And it has one of the largest broad host range of plant viruses. It can infect over 1,000 plant species, and some of the commercial vegetable hosts include lettuce, endive, radicchio, capsicum, peas, tomatoes. And it also can infect a lot of weeds across very different species. In terms of transmission of tomato spotted wilt, it's transmitted by 3 species of whitefly, but the western flower thrips is the most efficient in transmitting this virus. The life cycle of thrips from egg to adult take 20 to 30 days, but this is wholly dependent on temperature. When you have a higher temperature, you have a shorter life cycle, and this might lead to outbreaks that are observed during warmer months in lettuce production. The thrips acquire the virus through feeding on infected plants in the pupa stage, and once the thrip acquires the virus, it can transmit the virus throughout its life, and that's why it's efficient in transmitting the virus and can wipe out whole blocks of lettuce, as you can see here, a heavily infected block of lettuce. In terms of symptoms associated with tomato spotted wilt, they vary according to the host and also the variety, similar to other viruses. But generally, we see leaf yellowing, distortion, and necrotic spots, which is a characteristic leaf symptoms in most vegetables. And early infection in lettuce results in severe stunting and no formation of the head. We see purple or bronzing coloration of leaves in infected plants that are infected in early stages, and also ring pattern are common symptoms of tomato spotted wilt. Early infection also causes wilting and eventual plant death. So tomato spotted wilt is a very important virus for many vegetables in Australia, including lettuce, and it's the major issue in Australia where we've seen massive outbreaks in lettuce crops through this area-wide management project. Next, I'm going to touch on virus disease management, and generally all these viruses have a similar management criteria, and it is one of the most important things is to remove the source of the virus and the vector. So it's very important for you to control weeds. As you have seen through all the viruses, weeds are the most important reservoir or alternative host for the viruses. And there's this study done in 1963 where it showed that disease incidence reduced more than 70%, increased to more than 70% with no control of weeds, and there was a reduction, good reduction with control of weeds. So another thing which is important also is removal of or plowing under of leftover crops after harvesting. This is to reduce crop to crop production, but it's not applicable to some of the other viruses, such as lettuce big vein viruses, where crop debris which is left over can result in further infection of newly established crops. Another important thing is avoid contaminated planting materials such as seed or seedlings, which is another method which can introduce disease into your field. Also, there's integrated approaches such as vector monitoring, which is really important. It's good to know what vectors are in your field, and using maybe sticky traps, and which can inform you what sort of viruses that's causing diseases, because sometimes some of these viruses occur as mixed infection, and you can't identify them by symptoms alone. Other methods for controlling viruses are cultural methods such as crop rotation, planting schedules, and general crop hygiene and husbandry. And also there is use of biological controls where appropriate chemicals are not available, and also using of resistant varieties to some of the virus or even some of the vectors that transmit viruses. And for easier management of viruses, it's critical, first of all, to have reliable diagnostic. You have to know what you're up against before employing some of these management criteria. And in most states in Australia, I think all the states in Australia, we have diagnostic services offered by state agencies. And this is a good tool for growers to use in case they have an unknown disease or something they suspect is a virus or they're not sure what it is. I think that's the end of my talk, and I'd like to thank all these groups and Horticulture Innovation Australia, AusVeg, and thank you for listening.
Excellent, thanks for that, Cliff. A really great overview of some of those 4 key viruses in lettuce, as well as some really practical management approaches there. We've now got the chance to ask those burning questions from the audience of Cliff. So as I mentioned before, type those into the question pane and they'll come directly through to us. Cliff, just while we're waiting for people to send through those questions, control of lettuce big vein virus specifically and its vector, I mean, are there chemical control options available for that particular virus?
For the lettuce big vein virus, there have been trials to see if fumigation can be able to control the fungus that transmit the virus, but it's not that successful, and also it's very expensive. So it can reduce the titer of or the population of fungus in the soil. But I think other cultural methods such as growing, rotating with biofumigants can be able to reduce the level of fungus in the soil, which can be effective in some ways, but not entirely clear the fungus in the soil.
Yeah, great, thank you. You had a slide on the lettuce necrotic yellows virus there, just around difficult control with aphicides because it resides in the heart of lettuce. Are there specific management tips there because of where that resides and the difficulty getting in there with aphicides that you would suggest, some other cultural or perhaps biological control options?
I think biological methods using maybe predators, that might be the tool here. But I think the first thing is just preventing the aphid getting into your crop, and that's through clearing weeds and just general hygiene of your crop. But there's some studies that have found that using some of the biological predators, which can be effective in reducing the population of the lettuce aphid.
Great, thank you. And On the management options there, you touched on removing the source of the virus and vector and the importance of hygiene and good biosecurity. The points there on an integrated approach, as a grower starting out, what would you recommend the top 2 or 3 tips for them in starting to implement an integrated approach to managing these types of viruses?
I think the first one is on weeds around the property and between blocks of crop, because you can see most of these viruses, I think most of these insect vectors, aphids and thrips, most of the time they prefer the weeds to your crop. And if there are no weeds, they'll migrate to your crop. So the most important thing while you're trying to establish your crop is, first of all, to make sure that it's free of weeds, and continuous monitoring to know what vectors are in your block of land. I think those 2 are the most important things to consider.
Yeah, great. And monitoring, I mean, again, starting that out can sometimes be difficult, knowing which is a pest and which is a beneficial. You mentioned the diagnostic services. Are there other resources that you would recommend for monitoring insect pests, particularly aphids and thrips in this case as vectors of those diseases?
Yeah, I think if the diagnostic services are not available, I think most agronomies have enough information, that's what I believe, to be able to identify what insect vectors are on these traps. And I think also from our project, there were some outputs on some of these insect vectors, which can be as a reference guide to be able to compare and see, okay, this looks like this aphid. This might be what's causing, maybe might be a problem or might cause a disease, bring a disease in my block.
Yeah, great. We've had a couple of more questions come in. One from Sylvia here, just asking if you'd found any new or emerging viruses during the course of that area-wide management project that would be of concern, specifically for lettuce.
For lettuce, not really. We haven't found anything that's new emerging. It's just what we found for lettuce, just a variation in some years. It was a 4-year project. In some years, we found a high incidence of certain viruses, and sometimes it's a variation of seasons, but we haven't found any new viruses that might be causing an issue, apart from tomato spotted wilt, which is just a constant issue across lettuce production.
Yeah, thanks, Cliff. Another question from Carl, not myself, another one, just asking, have or do regular beneficial host rows within the crop, are they shown to provide sufficient disease control in that they're able to harbour enough beneficial insect populations?
Yeah, I think it's important, and I think there's a separate project around where they grow natives to try and build the beneficials population. But I think it's not just one tool that's effective. You need to combine several methods, having beneficials and having other cultural methods such as rotation, and also good crop hygiene. I think several approaches seem to work better than just one single approach.
Yeah, great. Thank you for that. And just while we're waiting for others to send them through, we've covered your top tips for an integrated approach, Cliff. With removing the source of the virus and the vector, quite often it's said in biosecurity you're only as strong as your weakest link, which can sometimes include your neighbours. What have you found during the project, particularly with these viruses, in, say, managing borders of paddocks or those that might abut public land? Where do growers and advisors start to have that discussion with neighbouring properties?
I think this is something we saw in some properties through this project, that maybe a vegetable grower is neighboring a flower farm where certain insect vectors are not a problem, but it's a huge problem for the vegetable grower. And I think that's why our project was trying to push the area-wide management, that managing this pathogen is not on a single farm basis, but in larger regions, and there should be conversation, and we were encouraging conversation between growers within a region to try and employ similar cultural methods in trying to put the population of insect vectors or alternative hosts down. And I think this is something important and a conversation that we should keep on stressing about, because it's just one grower within a region of many growers not doing the right thing, or not doing the correct thing in terms of management of weeds and other things might cause issues for a larger area. So I think it's just a conversation that needs to keep on going between groups of growers within a region to try and manage pests and diseases within a larger area than in single farms.
Yeah, some really good pointers there. We've had another question come in, Cliff, just around big vein. You mentioned during the presentation that can actually last in the soil for up to 25 years. A question from someone's just asked, with managing crop residue and reduced tillage, I mean, balancing that broader soil health versus virus management, what are some of the trade-offs there that you're going through from a decision-making point of view?
I think that's a very good question, because it's important also to manage the soil health, and some of these crop residues are important to building up the organic content of the soil, and I think a good way of managing big vein disease is use of resistant varieties, which currently there are several resistant varieties, and this can help you manage the virus, as well as also if cultural methods such as building your soil organic content is important, there's still continuity in your production without the heavy impact of the virus.
Yeah, great. No, thank you. We've probably got time just for one more. Another question has come in just with another La Nina year being announced by the Bureau, the third year in a row, and more moisture and heavy rain tipped for this summer. What should growers be looking out for, particularly around virus management? Is there anything else that they should be aware of, Cliff?
I think with rain, more weeds and more vectors. But the funny thing is sometimes also the rain suppresses the vectors, as they can't move as efficiently as normal because of the rain. But I think it's just weed management, because there's a lot of weeds out there, and when there's clear skies, the vectors will try and move from the weeds to the crop. So I think it's mainly it's just weed management that growers should look out for, and yeah, most of the and also virus transmitted by insect vectors, which I think when you have lots of weeds, it's something that we need to look out for.
Yeah, great. So we're coming to the end of our very short half-an-hour session this afternoon. Some really great takeaways there around good farm hygiene and crop management, also managing and removing weeds, and the importance of monitoring and good diagnostics as well. This is the third of 3 sessions in our area-wide management mini-series. If you've missed the other 2 on curcubits and capsicum, you can catch up on those at soilwealth.com.au and watch the recording and access the presentation, along with a host of a whole range of other virus resources and also broader soil management and plant health tips and tools. If you haven't already, please feel free to download those resources in the handout pane there, including those 2 great identification and management guides of bacterial and viral diseases. A big thank you to Cliff Kinoti from Agriculture Victoria for your time and expertise and sharing that with us this afternoon. We really do appreciate that, and thanks to you for getting along. Any other questions, please sing out, and we'll see you at the next Soil Wealth ICP event. Thanks everybody, bye.
Thank you.